Happy New Year, everyone. And this is the first session of our ISCB-SC RSG-India 2026 session. And this is we are continuing our EduSearch series that we have partnered with EMBL-EBI. And we are explaining the PDBe that we are doing from the past year. And the past year, we have successfully completed our 2 episodes. So right now, we are in our 3rd episode. And today, we have our new secretary of ICB-SC RSG India, Harshidi from Tamil Nadu, and she is going to introduce today's session. So, Harshidi, over to you. Harshidi.
Good evening, everyone. I am Harshidi, currently serving as the president of Tamil Nadu Node. We are proud to welcome you all to the first event of 2026 organized by ISCB-SC RSG India in collaboration with EMBL-EBI for the EduSearch series with PDBe-KB. This is episode 3 of Exploring the Protein Complex Resources, curated by PDB from Germany in EMBL. A brief introduction about our organization. The International Society for Computational Biology Student Council, Regional Student Group India. We are your local window to global opportunities in computational biology. ISCB is a global organization based in the USA, and it drives computational biology and bioinformatics forward through research, training, outreach, and community building. The ISCB Student Council, started in 2006, nurtures the next generation of computational biologists with RSGs in over 26 countries. Regional Student Group India connects and promotes a strong nationwide network of computational biologists linking local talent to global opportunities. We proudly collaborate with premier institutions including Mahindra University, NIT Calicut, Manipal Academy of Higher Education, IIT BHU Varanasi, and Sri Ramachandra Institute of Higher Education and Research. Here is the RSG India Core Committee for the year 2026, supported by faculty advisors and visionary leaders across India. RSG India has successfully organized impactful events in the past, fostering collaboration, learning, and innovation in computational biology. Here are pictures from some of the events organized from the year 2023 to 2026. That brings us to the end of the introduction of our organization, and I would now like to invite our guest,
Today's speaker, Dr. Sri Devan Appasamy. So, I will introduce him first. So, he's a bioinformatician at the Protein Data Bank in Europe, where he works on making macromolecular complex data more accessible to the scientific community. He completed his master's in molecular biology at University Kebangasan, Malaysia, and PhD in biology at Bowling Green State University. During his graduate studies, he developed computational tools for the annotation and analysis of RNA 3D motifs. At PDBe, his work focuses on building and improving the PDBe-KB complex resource, integrating structural data and biological context to support large-scale exploration of macromolecular complexes. So thank you, Dr. Sri, for coming to our platform and for giving this lecture. Over to you, sir. I'm just stopping my screen, and then you can share your screen, sir. Yes, sir, you can share your screen. Yeah, it's visible, sir.
Okay. Okay.
Yeah.
Okay. Can you see? Okay. Good. Perfect. All right. So good afternoon. Thank you for the kind introduction. So my name is Sri, and I'm a bioinformatician at PDBe, which is based in EBI. So I primarily work with macromolecular complexes, and with the goal of making the data more accessible to the scientific community. So today I'm going to talk about a resource that we recently developed called PDBe-KB Complexes. So my colleague Joseph has presented about PDBe-KB extensively, so I'm not going to repeat the same thing again. But I just want to just to bring your attention to this slide and to look at several these aggregated views. So we basically have developed several views, aggregated views. So at the moment we have views for proteins, ligands, and complexes. So these are already live in production, and for domains and nucleic acid are planned for future developments. So today I'm specifically going to talk about the aggregated views for complexes. All right, so I've broadly divided my presentation into three sections. So first, I will talk about the motivation and the background behind this work. And secondly, I'll talk about how to search and browse complexes data on PDBe website. And finally, I just have a small case study to see how we can go from prediction in AlphaFold DB to macromolecular complexes in PDBe-KB complexes. Disclaimer. So what is our definition of complexes here today? So here, so we adopt an inclusive definition of complexes covering PDB assemblies from monomers to large multi-chain assemblies. So this allows us to capture the ligand bound states and partial assemblies in addition to biologically relevant oligomers. So, the broad scope avoids excluding potentially informative assemblies and also provides a consistent basis for aggregation and comparison across the PDB. So of course, this depends on, because different people use different definitions for complexes. In standard definition, people consider assembly that consists of at least two chains to be a complex. But in our case, we consider even like monomers to be a complex. That's just something to take into consideration. So what is the motivation? So PDB assemblies in PD often lack a name and clear biological content. So previously, if you want to search for a particular you have to either rely on the structure title or the description of the individual macromolecule that makes up that complex. So there isn't a name that is assigned to the assembly itself. So if you want to search for assemblies in the website, it's not very reliable. So this makes that searching for a particular complex difficult on PDBe website. Additionally, there's a lack of systematic aggregation of assemblies at a complex level across the whole PDB archive. You might ask, why is it important to aggregate all the assemblies in the PDB? So in PDB, the same protein might be solved multiple times under different conditions. In one case, you might have the same protein bound to a different ligand. It can be bound to maybe a different set of macromolecular partners. And people might also like change the pH or introduce mutations, modifications, and so forth. So with that, one can ask the question: so how does these modifications of bound ligands influence the structure and the function? So the answer to this question is rarely answered by looking at single structure alone. So that is the value of the redundancy in PDB archive. Like why is it important to do this? And then of course you can ask the question, which PDB assemblies represent the same macromolecular complex? What are the subcomplexes and supercomplexes of a given complex? So this makes it very difficult at the moment to study variability among instances of the same complex. It also hinders the analysis of relationship between a complex and its macromolecular partners. So that was primarily the motivation for us to build this service. So there are some excellent resources for macromolecular complexes. So I've listed some of them here. So the first one is Complex Portal. So this is actually a database that is also hosted at the European Bioinformatics Institute. So it is a manually curated database of macromolecular complexes from model organisms, but without systematic structural coverage of the PDB. So this is more like a functional database containing functional annotations. Similarly, Quorum is a mammalian protein complex curated composition with biomedical annotation. So both these databases are similar in the sense that they are functional databases and don't really look at the structural aspect. And then there's database called 3D Complex. So this is a structure-oriented database that classifies protein complexes by sequence similarity, structural features, and contact topology. So this database is very useful if you want to investigate, for example, complex topology, and you want to study topology between different complexes. So this would be a very useful uh, resource for such tasks. And then you have the other database called ProCat. So this is also another structure-based resource which clusters recurring biological protein assemblies across independent PDB experiments to assess biological relevance. So this is in a bit similar to what we are doing, but here they are just focusing on protein assemblies and also cross-species clustering, whereas we are only looking at within the same clustering within the same species. And then there are some specialized databases like Ribo XYZ and GPCRDB that basically is a domain-specific databases for ribosomal complexes and G protein coupled receptors, offering curated annotation and structure-focused tools. So these are great. I mean, if you're interested in a particular complex, but looking at all these different databases, they don't really give you aggregation across PDB archive in a systematic manner. So next we look at the foundation for the complex pager. So in PDBe-KB, we aggregate structural data around specific entities. So for the protein pages, based on what Joseph has shown previously, we basically group this based on the UniProt accessions. And for the ligand pages, we group them based on the chemical component identifiers. So as part of this work, we have developed an aggregated view of macromolecular complexes. The work is based on several following prerequisites. So first, we need to aggregate assemblies that represent the same complex. Secondly, we need to assign unique identifiers to distinct complexes. Third, we need to generate persistent identifiers so that they stay stable over time, and then you will be able to track any changes to those identifiers, something changed. And finally, whenever possible, we want to try to name the complexes in a human readable way. So here I'm going to explain to you how we did the aggregation workflow. So for all assemblies, we first identify all components in an assembly using accession identifiers whenever available. So if it is a protein component, we map it to UniProt. If it is an RNA component, we map it to RNA to RFAB. And then we identify the stoichiometry of each component. So taking the human hemoglobin, for example, where we have the alpha subunit and beta subunit, and each subunit exists in two copies. So for components that cannot be mapped to either UniProt or RFAB, we create a label that uses a combination of polymer type, PDB ID, entity ID, and stoichiometry. So I will show this in the next slide. Then we basically aggregate the assemblies based on the unique set of the components. And then we also exclude some short molecules during this aggregation. So the idea here is that in some PDB structures, you might have short nucleic acid fragments or antibodies or peptides. So these are not the core component of the protein. So in that way, so we basically excluded them so that we only focus on the components that make up the core of the protein. And then we basically assign persistent identifiers for each unique composition. And then finally, we assign human readable names to each unique complete composition. This is done automatically, but also with some manual curation involved. All right, so how do we generate the labels for this assembly component? So just like I said earlier, for something that can be mapped to either UniProt or RFAB, it consists of only accession and stoichiometry separated by an underscore. And then we have other classes. So, antibody is a special class of protein. So we use a tool called Anaki to annotate antibody chains in the PDB. So this is a tool that was developed by the Charlotte Dean's group at Oxford. So it is actually a standard and a widely used tool for annotating antibody chains in the PDB. And then we have a protein that is unmapped. So these are basically a protein component that cannot be mapped to UniProt. And then we have RNA unmapped. So these are RNA chains that cannot be mapped to RFAM. For DNA, we don't have any external data. because we don't have it in terms of family databases, and also usually most of the DNA sequences that you have in PDB entries are quite short. Not always, but the majority of them. So we don't map DNA to any external resources for now, at least. And then we have another category for DNA RNA hybrid. And then we have some additional components. So these are the components that we exclude during the aggregation process. So basically, we exclude both mRNA and tRNA in ribosomal complexes because these are usually more like ligands that bind to the ribosome and not really the core component of the ribosome. And then we have peptide. So we consider any protein chains less than 20 amino acids as peptide. And then we have different categories for nucleic acid fragments, RNA, DNA, DNA-RNA hybrid. And these are basically less than 25 nucleotide in length. And we basically add a SH prefix to make a note that it's just a short fragment. Okay, so with that, I just want to show you some examples of some complexes and their assembly composition. So first we have an example of a complex called tRNA methyltransferase, and then we have the composition label. I mean, just like by looking at the composition label, you can get a good idea what it's made up of. So in this case, it's a heterodimer and made up of two different components. And then these are basically all the PDB assemblies that represent this particular complex. And then we have a collagen trimer. So this is basically a trimer, a homotrimer. And then you have another complex here, another example, methionine ABC transporter complex. So if you look at the composition label, you can see that it is a pentamer. Since you have the total stoichiometry is 5, that is made up of 3 different components. So just by looking at the assembly composition of the complex, how many components you have, and what is the stoichiometry of each component that makes up that particular complex. Okay, so this is basically our workflow on how we name complexes. So we rely on several. So the problem is that, like I mentioned earlier, so the current annotation practices lack standard naming conventions for complexes in the PDB. So we do not name assemblies in the PDB. We only name, we have the structure title, and then you have the description for the individual macromolecule. So here is our strategy basically, So we use, our gold standard reference is complex portal because this is a manually curated database. And secondly, we check whether if it doesn't have, we don't have mapping to complex portal, we check whether it is UniProt, mapping to UniProt. The majority of complexes are named using UniProt because these are usually monomeric assemblies or homomeric assemblies. So in such a way, you can just transfer the name from UniProt to the complexes. So basically, UniProt forms the bulk of the naming category. So in addition to that, we also have several different ways. We also have like a manually curated data set, but this only involves a small number of complexes. In addition to that, we also use Go ontology, where we check whether all of the components, all of the macromolecules, or all the components have a common Go ontology names. If they have a common names, then we try to use that. And then we also have like some specialized code to name ribosomes. So basically, the big picture is that we use resources external to PDB, such as complex portal, UniProt, gene ontology, RFEM, and manual curation to name these complexes. So one important, so this name, it is not meant to be the ground truth, but it's just we've just assigned a consistent label to the complexes. And as you know, in biology, the most difficult thing is naming stuff, and that also applies for structures. So this is a evolving thing, and hopefully we can do better going forward. Okay, so now I want to talk a little bit about data availability. So this process is integrated into PDBe weekly release process to ensure that all new and updated PDB entries are assigned persistent complex identifiers. So using our process, we were able to aggregate all of PDB assemblies into over 100,000 unique complex records based on their composition. So this is as of November 2025. and we were able to also assign names to almost over about 90% of them. So how could you access the data? So there are three primary ways how you could access them. The first would be through the FTP side. So at the FTP side, you can basically find multiple CSV files containing data related to complexes. There is also a README file in the FTP side, which basically explains what kind of information is available in each of the CSV files and what does the different columns represent in the CSV file. So that would be really helpful for you to help to interpret the data, the CSV files. And then we also have APIs, which I will show in the next slide. And then you can also use the PDBe website to access this data. And we also have a Jupyter notebook that has a PDB-wide exploratory analysis of complex data. So if you're interested, you can just git clone the notebook and use it for your work. Maybe it should be pretty straightforward. And so this is useful because if you want to like analyze the composition of the complexes, you want to know what are homomeric assemblies, what are heteromeric assemblies. So this would be a useful notebook for something that you can use. Okay, so I'm not going to go this into detail. So I'm just going to describe the different endpoints that we have. So here we basically have 4 different variants of the same endpoint. It's just that we query it using different identifier. So the first one is something called PDB complex ID. So we basically have PDB complex ID for each assembly in the PDB. So how can we find this PDB complex ID? First, you can look at the CSV file in the FTP site. There is a master CSV file that contains the mapping between each individual assembly and also PDB complex ID. And then this PDB complex ID is also available on the PDBe website, which I will show in a short while. Secondly, you can also query using accession. So this is useful if you want to identify all the complexes that contain a particular protein component or RFAM component or RNA component. So accession here means that you can either use uniprot accession or RFAM accession. And thirdly, you can also query this endpoint using PDB ID. So why is this useful? So remember that a PDB entry can have multiple assemblies. And sometimes, these assemblies have different composition. So it is entirely possible that you have multiple PDB complex IDs with PDB entry. So basically, for each unique composition, you would have a unique complex identifier. And finally, you can also use complex portal ID. So if you're someone that uses complex portal ID and you're interested in a particular complex, you can also use that ID to see whether that particular complex exists in the PDB database or not. And then there are also some additional endpoints that you can use to query ligand details, subcomplexes and supercomplexes information, and also predicted PISA assembly parameters. Okay. So here I just have a few short figures showing just some analysis today. So number of complexes. So unsurprisingly, protein forms the bulk of the complexes in the PDB. So when I say complexes here, I mean the unique complex compositions. So almost close to 90%, 88% are just protein-only complexes. And then you have another remaining 7%, which are protein nucleic acid complexes. And the final 5% are basically just nucleic acid complexes. So this figure shows the experimental methods used to solve complexes in the PDB. Again, not very surprising, the bulk of complexes are solved using X-ray. But you can see that EM slowly, so about 15% of the complexes in PDB are solved using EM. Again, the interesting thing here is that you can see that there are some complexes where the constituent assemblies were solved using a combination of methods. For example, there is NMR and X-ray were used to solve about 2% of complexes, and about 1% of complexes structures were solved using a combination of EM and X-ray. Okay, so here I'm just showing you how the distribution of complex named sources. As you can see, uniprot forms the largest naming category, or almost 60%. And then we have heterodimers, almost 10%. And then we have another category where we have all unmapped proteins. So these are basically complexes where the individual component cannot be mapped to UniProt. So that's almost 10%. And also we have about close to 10% where we cannot assign the name. So these are usually the large heterometric complexes where we can't depend on any automatic method to name them. Right. So what we are trying to do now is that we are getting more and more larger complexes. So it's just not possible to like manually curate the names for these complexes. But in the recent years, there's been a lot of advances in these large language models. So one thing that we are trying to explore is that how can we use these large language models to extract names from literature and maybe from primary citations and name the complexes automatically. But that is still an evolving work and something that we're still actively exploring. Okay, so coming to the second part of my presentation today, it's just going to talk about how we can search and browse complexes data in the PDBe website. Okay, so the first I'm going to just show you the home page today. So you can use the advanced search to search for complexes data. Once you do that, it brings up this particular drop-down menu where you can search, you can basically type keywords and select the search field. So there are two different search fields that would be relevant for complexes. One would be the complex name, and the second would be the complex ID. So let's see what you can do. So first, you can search for assemblies using complex name. So for example, querying for hemoglobin in the complex name search field will yield, with the condition contains, yields multiple related assemblies. So here you have an example of a hemoglobin from alligator, and you also have an example of human hemoglobin bound to additional molecule from different microbial pathogens. So here you can use different conditions. So contents is more fuzzy search, but you can also use the condition equal to, or whether the keyword is present at the start of the search or at the end of the string, basically. So these results basically represent related variants of the same biological complex. In this case, this is useful if you want to search for hemoglobins across different species. So you might find them, in different stoichiometry or maybe bound to different macromolecular partners. So this kind of fuzzy search is very useful if you want to explore related assemblies and their biological variation across the PDB. Secondly, you can search for assemblies representing the same complex. So you can search basically by using the PDB, by typing the PDBe complex identifier. So this would basically enable you to identify all PDB entries containing a given assembly with identical composition and species. For example, searching for PDB complex ID, PDB-CPX 154652 will display all PDB entries that correspond to the adult human hemoglobin. So this is basically you want to find all instances of a complex from the same species. So you use the complex identifier. Okay. So next, I think I want to show a little bit about the entry pages and how you can access the complex data from the complex pages. I will try to play this video and see whether it runs. Okay, so we recently redesigned the PDBe entry pages. So if you've been using the PDB entry pages previously, so I think the major change that you would have observed is that we have moved to a tab-based layout, where each tab corresponds to different section of the structural data. You can look at macromolecules, ligands, and domains, and so on. So the idea here is that the central theme of this redesign is that to enable users to use the 3D viewer to explore the data in addition to the annotations that are displayed on the pages. So here I'm just going to show you an example of the complexes section in entry pages. So here we're looking at the spike protein complex. So basically, you have basically a link. This is the link to the aggregated view. And then, you have the complex name, you have the state, the oligomeric state, and then you have the PDB complex ID. So clicking on this will basically bring to the search page, which I've shown earlier, that lists all the PDB entries containing particular assembly. And then you also have some PISA parameters. Okay, so let's look at the aggregated views of complexes. So the pages are now live in production. So there are 7 sections now available: summary, structures, PISA, ligand, subcomplexes, supercomplexes, and citations. So I will use the spike protein as an example to walk through the different sections of the page. So you could also access these pages directly using the following identifiers. So you have the base URL, and then you just append the different identifier. It could be a PDB complex ID, a PDB ID, or the complex portal ID. Okay, just going back, I want to explain what do we mean by preferred here today. So in some PDB entries, there might be multiple assemblies in different composition. So for example, you might have a PDB entry that has a homodimer and a homotetramer, for example. So we define preferred as the assembly that has all the unique chains in the smaller stoichiometry. So in that particular example, the homodimer would be a preferred assembly. Now that's just an example that we use in PDBe to choose like a representative assembly for a particular PDB entry, just to clarify what is meant by the concept of preferred assembly here. Okay, so let's look at the summary section. So here we're looking at a spike protein. As you know, probably today, it's probably one of the most extensively characterized macromolecular complexes in PDB, especially during the COVID pandemic. So, when you look at it, what are some of the key things that might be obvious to you? We have a complex portal ID. So you can look at, if you want to understand more at the functional aspect of this complex, then you can go to complex portal. And then you have the components that make up this complex. So in this case, it is a homotrimer. And then you have something called global symmetry. So one thing that you can notice is that more than half of these assemblies here have a different symmetry. So cyclic C3 is basically the symmetry that is adopted by majority instances of this complex. So as you know, the SARS spike COV2 protein is a homotrimer, and each subunit has a domain called the receptor binding domain. So this receptor binding domain can be in up conformation or down conformation. So when the spike protein has all 3 receptor binding domain in down conformation, it basically adopts the C3 symmetry. But if you have 1 RBD up or 2 RBD up, that basically disrupts the C3 symmetry. So this is probably one of the ways that you can identify assemblies in different conformations. So this is one indication that you can use to see that, okay, this complex might exist in different arrangement of conformations. And then you have unique bound macromolecules. This just tells you that what are the unique bound macromolecules that are bound to this particular complex. So in this case, we have 2. We have antibody and peptide. And then you have the matrix overview section. So this tells you that what are the experimental techniques that are used to solve this particular complex. And you can see that for this particular complex, all 3 of the main experimental techniques were used, although EM is the most predominant technique used to solve the instances of this complex. Next, we can look at the structure section. So this basically just lists all the instances of this complex, and you can inspect the individual structure from here. And then you have the filter option here today. So you can filter by the bound macromolecule. So in the case, if you want to just look at instances that are bound to antibody, so you can choose from a drop down, you can choose the antibody. And here it will only show you the instances that are bound to antibody. So almost like close to 500 assemblies are bound to antibodies for this complex. And also that most of the columns are sortable and searchable, so it's very useful if you want to search for specific keyword in the title. You might want to search for open state or closed state or RBD up and RBD down. Once you have filtered the column based on specific keywords, and you can also download the CSV. And basically, the nice thing is that the CSV will basically contain the filtered data set and not the whole CSV, so that you don't need to filter it again after you download the CSV file. Next, we have the PISA section. So PISA is a program, I'm not sure whether many of you are familiar with this, but it's a program they use to predict assemblies in a complex and also to identify interfaces. So in this here, we are basically listing all the assembly properties for all instances of this particular complex. That is what each row in this table represents. We also have a filter where you can filter it by resolution or by experimental techniques. So the reason why we have this filter is that some of these values can basically correspond to experimental techniques or also the resolution that we use. Here, you basically have a slider that basically gives you the ranges. So you basically can inspect where the value for the individual assembly stands with respect to all instances of this complex. And also you have a histogram, which tells you the distribution of the values. So this is interesting because the outliers here, they may indicate partial fragments, incorrect annotation, or alternative confirmation. So we basically map the protein components to UniProt, but the one thing that we don't really check is the degree of completeness. So it might be entirely possible that you might have some instances that are truncated. So you will have to use all these visual cues in order to be able to deduce that. We are working on making some kind of metric to tell you whether the complex is complete or not. But at the moment, we don't have anything on these pages to tell you whether it is a complete complex or not. And then we have the ligand section. Basically, this just shows you all the unique ligands that are bound to the instances of this complex. So this is very useful if you just want a big picture of all the chemical context, of this particular complex. So, in the case of spike protein, we know that, it has a glycan shield. So it is unsurprising to see many of these chemical ligands are basically sugars, basically. And basically, there's also you can also sort by functional type. For the spike protein, we don't have any functional categories. But if you look at different complexes, there might be additional functional categories like drug-like, cofactor-like, or reactant-like. So we basically have a process that runs weekly that basically annotates a ligand whether it is drug-like, cofactor-like, or reactant-like. So that will help you to distinguish whether the ligands are something that's needed for the function of the protein, or are the ligands that are used for structure determination. Then we have the supercomplexes section. So here you can basically see what are the available supercomplexes for this particular complex. So a supercomplex is just defined as a complex that has additional components bound to the base complex. That's basically what it is. So when you basically click on the individual row, it will basically superpose the representative structure of the base complex and the representative structure of the supercomplex. That is what you're seeing here. So here I'm basically superposing a supercomplex that has bound to ACE2, angiotensin converting enzyme 2, compared to the base complex. So any additional components are basically colored by unique entities. So basically here, these are the ACE2, basically the unique entities here. Alright, so finally, we just have a citation section. So nothing much to say here. So these are basically just all the individual publications that are associated with the PDB entries that represent this complex. Okay, moving on. So just very quickly to say what is next for the complex pages. So what I've shown so far is just a very what we have done so far is just like the foundational work, to organize the complex data. So I've not shown you any annotations, any annotation from external databases. You might have seen this in PDBe-KB when Joseph has presented PDBe-KB, where we have all this annotation from external databases, but we don't have this for the PDBe-KB complexes yet. So that is because one of the biggest challenge with complexes is to define chain equivalence, because each each assembly might have more than one chain. So if you're looking at two instances of the same protein that have multiple chains, you need to be able to determine the chain equivalence. So that is what we are doing at the moment to identify equivalent interfaces, and this will then enable us to annotate and compare the equivalent chain across the PDB entries, and also to cluster complexes where you might want to just look at a particular subunit. You don't want to look at the whole complexes. You might just want to look at one particular subunit clustered across all instances of that complex. And one thing I'm also actively working on right now is to build a prototype for interfaces. So the idea here is that how to explore how interfaces vary across multiple instances of this complex. And we can use this to answer the question like, what are the interface differences between the different complex, and then we can link this interface variation or functional state. All right, so this is just a very simple example of prototype for the interfaces view. We have a 3D viewer. We have a table that lists all the instances of the interfaces. Then you have a context table, and then a Sankey diagram just to show you what are the key residues that participate in this particular interface interaction. So the key thing to make note here is that this is basically the same interface, multiple instances, same complex, same organism, and see what we can learn from this. All right, moving on, final section. Okay, bridging prediction and experiment for macromolecular complexes. So I'm just going to give you a very simple example of how we can go from from AlphaFold database to PDBe-KB complexes. So why structure prediction needs biological context? So many proteins lack experimental structure in the PDB. So AlphaFold DB currently provides monomer prediction. Of course, we have now AlphaFold multiple. There are now deep learning tools that can annotate complexes. I mean, not large complexes, but at the moment, for alpha, as far as AlphaFold database is concerned, you only have monomer predictions. At the moment, I think we have close to more than 200 million structures, and a biological function typically arises in oligomeric complexes. So we need to be able to link prediction to validated experimental assemblies. So how do we go from prediction to biological context? So we start with at AlphaFold database with the predicted monomer structure, and then we can use FoldSeq for rapid identification of experimental homologs. And then we can use the PDB entry page for validated experimental evidence. And finally, we can use PDBe-KB complexes to look at aggregated assemblies and biological contexts. So in this example, I've chosen a protein called the human shortwave sensitive opsin-1 protein. So there's no experimental human structure in the PDB. So mutations causes tryptopia, which is a color blindness disease. So AFDB provides a full-length monomer model. And if you see the domain annotation, so that stands for the Encyclopedia of Domains. So this was developed by the CAT team. This basically contains all the domains that are identified in the AlphaFold structured database. So the tab in this example identifies a 7-helix transmembrane domain with high PLDT scores. Next, we can use FoldSeq to retrieve the bovine rhodopsin homolog in the PDB. And we can see that we get bovine rhodopsins as the closest experimental match. So the top 10 hits share almost like 45% similarity, sequence similarity. between 2 to 3 angstrom resolution. The nice thing is that you can see that the 7 helix transmembrane domain that is very full is conserved among the experimentally determined structures. So this gives you some confidence that okay, this fold is it's a conserved fold experimental structure. Next, you can look at the X-ray structure from PDB. So this is we're looking at the PDB entry 30AX, which is a bovine rhodopsin structure at 2.6 angstroms resolution. So you look at the domain annotation. So this domain annotation by CAT supports classification as a rhodopsin family GPCR with 7 transmembrane helixes. So one thing to note is that this PDB entry has multiple assemblies in different oligomeric state. I'm just showing you the dimer here today. So next, you can explore PDBe-KB complexes. So as you know, I mean, if you go to complexes, I'm not showing it here, but you can directly link this to the complexes pages. So in this case, we have, this is a page for bovine rhodopsin dopsin. So there are 28 instances of the dimers in the PDB. So you also have some instances of this complex that have antibodies bound. And some of the key ligands include functionally relevant cofactors such as retinol and commonly occurring molecules such as sugars and lipids. So the idea here is that what I wanted to show is that we can go all the way from prediction in AFDB to the complex pages. So summary and outlook, predictions gain value when you link to experiments complexes, and PDBe currently enables seamless navigation from AFDB to biology, and PDBe-KB complexes adds a missing complex level view. So this is a scalable framework applicable beyond this case study. I mean, so you can use any protein that that you're interested in, and basically go all the way from PDB. I mean, from prediction to complex pages. Of course, the key idea is that there is an experimental structure for that particular predicted structure. If you have one, then definitely you can use it to be able to see whether you can get any insight into the function or the structure of that particular structure that we are looking at. So with that, I think that's all I have. So with this, I would acknowledge all of the team members at PDBe team and also our various grant providers. And I'm happy to take any questions. Thank you. Do I stop sharing?
No, sir. Yeah, thank you, sir, for your presentation. If somebody wants to ask some questions, they can write in the chat box. So there's a question there. So question from Nitya. So she is asking, would like to know if you consider the data of small proteins.
So if this data is available in PDB, I mean, so we do it for any structure. So if it is there in PDB, then it should be classified, it should be aggregated.
Any other questions someone like to ask?
Well, I saw a question about a guide from basic to bioinformatics. So that depends on what is your goal. I mean, is this more on about learning or about working as a bioinformatician? So if you can give me some context that I can explain more what would be useful. Yeah, so I think obviously now, like bioinformatician, you need to know what kind of skills that you need to have. And I think it's pretty standard, you must be good you need to know a programming language. At the moment, Python is the standard language. And then you must be comfortable using the terminal command line. What else? So also, I think mathematics would play a key role. So the reason is what, because nowadays you're going to have a lot of these machine learning things and stuff like that. Obviously, you can use the tools, but if you really want to master the tools, you need to understand the underlying mathematics. So I think nothing too fairly advanced. Linear algebra, probability, and probability, and also calculus. So I think this would be good skills to have. Yeah, but I mean, it really depends on how deep you want to go. And if you just want to use tools, then obviously you don't really need to be able to, to master this. But I think if you really want to be able to build tools and to make new discoveries using mathematically, then you will need to know this. Yeah, that's what I would say. Right. So that is a good question. So actually, so when I said earlier, so we have three aggregated pages. So I've talked about complexes, and Joseph has presented about PDBe-KB proteins. So that is more like single chain proteins. We also have aggregated views for ligands actually. So when you say structural descriptors, uh, so when you say descriptors, can you just tell me some example? What do you mean by structural descriptors? Are you talking about the interaction type? Is that what you're asking? So we in PDBe we use a program called RPGO to annotate protein ligand interactions. So if you go to the entity page and you go to the ligand section, you can basically look at protein ligand interactions. Okay, yeah. So I think at the moment we do not have this, but my colleague is actually working on clustering ligand binding pockets. So I think this might be something relevant to that. So his name is Roshan actually. If you go to the PDBe pages, you can look at the members. His name is Roshan, so you can directly ask him. So he's actually better placed to answer this question.
Is there any other question someone would like to ask? So there's a question from my side is that so
Sure.
if there is a structure, if there's a protein whose structure, basically when we find we have a sequence of a protein and we try to find the structure of that protein, we don't
Mm-hmm.
have the X-ray crystallograph, we don't have the X-ray crystallography structure of that protein in very well resolution, but we
Okay.
have an electron microscopy structure of that protein. So
Mm-hmm.
what is the different kind of should we go with that electron microscopy structure, or what will be the procedure for further our study? Or
Right. So I think it depends really what is your goal. So when you use for this EM structure, of course, the first thing that you would look at is resolution. And if
yes, sir.
something is, if it's less than 3 angstrom, it's fairly decent. But there's
Hmm.
also something called a Q score, which is actually a validation for cryo-EM structures. That will actually be available under the model validation in the PDB entry pages. So you can use that
Okay.
as a guide to see, how good is the structure. I mean, the structure. It might be possible that you might have a good resolution, but in some regions it's really bad. So it really depends, what region you're comparing, and So I would suggest is that first you look at the resolution, and then you look at the validation matrix. So that is what I would say. Because I think
Okay. Okay,
nowadays
sir.
most structures are solved using cryo-EM. So you really have a lot of
Yeah.
cryo-EM stuff. Even like the
Yeah.
spike complex, most of it are solved using cryo-EM.
Yeah. As your data is shown in the one of the slides is that 75% are X-ray, but the rest of them were
Right,
mostly from
right,
REM.
right.
Yeah.
I think those are mostly like the earlier structures. I think now
Okay.
people increase. I mean, I think that these large complexes are becoming, very like a routine structure. Everybody is solving large complexes using cryo-EM. Yeah.
Yeah. Okay. Okay. Thank you, sir.
Sir, all right, very much. Right. Thank you very much.
Okay. So I can share my screen then. So we have to share the feedback from sir. So can you stop? Yeah. Thank you, sir. So Janak, are you there?
Yes, I'm here.
Yeah.
Okay. So, yeah, thank you once again, sir, for the thoughtful and insightful master class. And that's a wrap for episode 3 of EduSearch series. But I would say stick around. We have got more exciting episodes coming up. Please do follow our RSG-India social media channels. We are on Twitter, Instagram, LinkedIn, everywhere. And check the ISCB website and the RSG India website also for updates and opportunities. So here's the feedback form. You all can fill it, and we are also open for volunteer entries. If you guys need to join us, you can fill out the volunteer form, and your skills make real difference. Finally, a heartfelt thanks to the team of EMBL-EBI, PDBe and PDBe-KB, and our incredible RSG India volunteers, our team. This wouldn't have been possible without you. Thank you, guys.